All right, let's jump right into this explainer and completely demystify the biological chain reaction behind systemic sclerosis. If you've ever looked at the pathophysiology of this condition, it can honestly seem like just a chaotic jumble of immune responses, vascular issues, and skin changes. But actually, it's a highly structured sequential process. Today, we're going to map out the pathophysiology of systemic sclerosis by looking at it like a precise sequence of falling dominoes.
To make this explainer easier to follow, we're breaking it down into 4 distinct parts. First, the genetic foundation, then phase 1, which is vascular damage, phase 2, covering immune system chaos, and finally phase 3, systemic fibrosis.
Section 1, the genetic foundation, setting the wobbly table. So, before a single domino can even start falling, we have to look at the foundational vulnerabilities. This disease doesn't just appear out of nowhere, right? All the downstream disease phenomena are explicitly anchored to a genetic base. Everything we're about to discuss physically builds on top of this invisible foundation. And understanding this baseline is absolutely crucial because it explains exactly why certain people are susceptible to this entire cascade happening in the first place. Let's clearly define this so we're all on exactly the same page. A genetic predisposition means the body is carrying inherited biological vulnerabilities. To use our analogy, the patient has a wobbly table. The table hasn't collapsed yet. The disease hasn't actually started, but the structural integrity is definitely compromised. It's highly vulnerable to a triggering event. So, what's actually making that table wobble? Well, it comes down to human leukocyte antigen susceptibilities, specifically the markers HLA-DR2, DR3, and DR5. There are also reported familial cases, which reinforces the inherited nature of the disease, along with a TGF-B receptor polymorphism. Now, without getting bogged down in the deep molecular biology, just know that these genetic quirks are the primer. They're the reason that very first domino is balanced so precariously.
Moving on to section 2, phase 1, vascular damage. The first domino falls.
The exact starting point for the active disease is endothelial cell activation. The endothelial cells are the inner lining of your blood vessels, and when they become inappropriately activated, it leads directly to the development of microangiopathy and vasculopathy, or, in plain English, small blood vessel disease. The actual piping of the circulatory system starts to fundamentally break down. To really understand how these vessels get damaged, we need to look at a critical term, capillary rarefication. What this means is there's an anarchic or totally chaotic proliferation of the intima and media cells. Those are the cells making up the walls of the blood vessels. They start growing out of control, inwards. And this physical overgrowth dangerously reduces the vessel lumen, meaning the actual open space inside those microscopic tubes where the blood flows just gets narrower and narrower. Now, the real kicker is that when these vessels physically shrink and get choked off, the surrounding tissues are completely starved of oxygen, a condition known as hypoxia. This lack of oxygen, combined with the cellular damage, triggers massive oxidative stress phenomena. Think of this like the damaged tissues pulling a biological fire alarm. The blood vessels are suffocating and they're sending out a desperate SOS to the rest of the body.
Which triggers section 3, phase 2, immune system chaos. Answering the alarm.
That biological SOS from the suffocating blood vessels is physically transmitted by the distressed endothelial cells. Remember, those are the cells lining the vessels. Well, they start increasing their expression of adhesion molecules. You can kind of think of these adhesion molecules like microscopic strips of Velcro appearing on the inside of the blood vessels. They're specifically designed to grab onto passing immune cells and recruit them right to the scene of the damage.
But once those immune cells get snagged by the Velcro and pulled into the tissue, things really go off the rails. These recruited immune cells begin secreting inflammatory and pro-fibrosin mediators. I want you to imagine these mediators as faulty chemical blueprints being handed out at a cellular construction site. Instead of fixing the damaged blood vessels, the immune cells are handing out orders that essentially say, hey, cause inflammation and start building scar tissue. And there's another really fascinating wrinkle here. Humoral immunity is deeply involved. We know this because most patients with systemic sclerosis carry autoantibodies in their blood. Now, their exact pathogenic role, meaning exactly how these autoantibodies directly cause the damage, isn't clearly established just yet. But their presence proves that the body's immune system has fundamentally lost its ability to tell friend from foe. It adds a massive layer of complex chaos to this whole cascading response.
Okay, let's just pause for a quick second and track our dominoes, because this sequence is the key to understanding the entire explainer. We started with the wobbly table of genetics. Then, domino 1, vascular damage causes endothelial cell activation. Domino 2, those cells put up adhesion molecules which recruit the immune system. Domino 3, the immune system arrives and hands off those faulty, pro-fibrosing blueprints to the cells in the tissue. And this precise sequence sets up our final devastating phase. Section 4. Phase 3. Systemic fibrosis. Overbuilding the scaffolding. So, this brings us to the definitive clinical endpoint of the disease: skin sclerosis and systemic fibrosis. The immune system handed out those faulty blueprints, and the cellular builders, well, they've taken them entirely literally. The result is the physical hardening and thickening of the skin, and eventually the internal organs. The primary culprit in this final stage is uncontrolled fibroblast activation. Fibroblasts are the normal cellular builders in your body. They're responsible for maintaining structural integrity. But stimulated by those pro-fibrosing mediators from the chaotic immune system, these fibroblasts go completely rogue. They become hyperactive, uncontrolled microscopic builders, aggressively oversynthesizing extracellular matrix. They literally just keep laying down collagen and structural proteins until the tissues completely stiffen and harden, losing their normal function. And that brings us to the end of the line. By stepping through this explainer, we've successfully mapped out the pathophysiology of systemic sclerosis, from a wobbly genetic foundation to blood vessel damage to immune chaos, and finally to stiffened fibrotic tissues. Because we now understand this as a precise 1-2-3 sequence, we have to ask a final provocative question. If this is a highly predictable chain reaction, where is the ultimate point of intervention? Do we try to fix the vascular hypoxia early on, or do we try to calm the chaotic immune cells? Or do we try to stop the fibroblasts from building? Understanding this cascade is the first step, but finding the right domino to pull out of the line, well, that's the true key to finally stopping this incredible disease process.